Hello friends, welcome to CEC Live Lectures. Dear friends, today in this session we are going to talk on one of the most interesting topics, and the topic of discussion is understanding the immune system. We are going to discuss how immune system works, and for the discussion on the topic we have with us in our studios Dr. Amit Bhattacharya. Dr. Amit Bhattacharya is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Dr. Amit Bhattacharya is a prolific professor. He persistently and constantly makes effort so that the student community is benefited as a whole. So friends, let's take advantages from his experiences and let's try to understand today's topic in detail, that how does the immune system work. Hello sir, welcome to the lecture.
Thank you very much, ma'am. Dear friends, in this part of the lecture I will be talking about how exactly the immune system builds up and what are the various cells which forms the immune system, and subsequently we'll talk about the various antibodies which functions in these immune systems. Now, the word immunity actually means the state of protection from infectious diseases. So, in this representation, if we can see, there is a macrophage which is interacting with the bacteria, and how exactly these macrophages engulfs these bacteria and they are cleared from the blood circulations. Now, the various types of cells of the immune systems, which are majorly the leukocytes, have been classified into various categories. The first one is called as the neutrophil, which constitutes about 50 to 70% of the leukocytes. Then comes the lymphocytes, which constitutes to about 20 to 40%. Then the monocytes, which constitutes to 1 to 6%. Eosinophil, which constitutes to 1 to 3%. Basophil, which are less than 1%. Along with this leukocyte, we have in our blood the red blood cells and the platelets, which plays a very important role in the other body functioning. Now, the organs of the immune systems have been broadly classified into two major categories. One is called as the primary lymphoid organs. The second one is called as the secondary lymphoid organ. The primary lymphoid organ, it provides the appropriate microenvironment for the development and maturation of the lymphocytes. The primary lymphoid organ are basically, they are the thymus and the bone marrow. So these are the organ where the development of the immune system cells generally takes place. The secondary lymphoid organs are specifically the places where it traps the antigen from the defined tissues or the vascular spaces, and these are especially the sites for the maturation of the lymphocytes that can interact effectively with that antigens over there or the foreign particle. Now, the blood vessels and the lymphatic systems connect all these organs, uniting them to one another. So, the primary lymphoid organs and the secondary lymphoid organs, along with the lymphatic vessels, they form the immune system mechanism in the human. Now, in the humans, one of the most important immune system mechanism which functions is called as the hematopoiesis. So, hematopoiesis is basically the formation and development of red and white blood cells. In this, hematopoiesis basically takes place under specialized cell or microenvironments which are present in the bone marrow. In the bone marrow, the red blood cells are formed, while the white blood cells are formed, which are broadly classified into lymphocytes, monocyte, eosinophil, basophil, neutrophils, and subsequently the platelets are formed, which helps in the blood clotting also. All these red blood cells, white blood cells or the leukocytes along with the platelets are formed from specialized type of cells which are called as the hematopoietic stem cells. Now, the hematopoietic stem cells are basically these specific stem cells are present at the site which is called as the human bone marrow. So over here we can see an image of the cross section of the bone marrow, where we can see the cartilage, spongy bone, the compact bone is showing over there, Along with that, there are bone marrows which are present, in which we can see the hematopoietic stem cells along with the stromal stem cells. So these stromal stem cells actually provides the nutrients for the and proper environment for the growth and the development of the hematopoietic stem cell. So human bone marrow cells plays a very important role in the induction and subsequent maintenance of the immune systems within the human cell. Now, if we see the electron micrograph over here, this shows a hematopoietic stem cell. So this is an electron micrograph image which shows the various types of the blood cells that are produced by the hematopoietic stem cell. So hematopoietic stem cells are basically, they are termed as the multipotent or pluripotent cells which are capable to differentiate into various ways, thereby generating the erythrocytes, granulocytes, monocytes, mast cells, lymphocytes and megakaryocytes. These stem cells are very few in number, and it has been found that only one hematopoietic stem cell per 50,000 cells is present in the bone marrow. Now, if we see the growth and the formation and the development of the red blood cells and the white blood cells, we can see that the hematopoietic stem cells, which have got a property of pluripotency and multipotency, they can self-renew themselves. Along with that, they can form into two major lineages. One is called as the myeloid progenitor cells, the other one is called as the lymphoid progenitor cell. The myeloid progenitor cell subsequently goes into the formations of the dendritic cells, monocytes, leukocytes, eosinophil, basophil, platelets and the erythrocytes, while the lymphoid progenitor cells develops into the major cells which helps in combating the foreign particles, which includes the natural killer cells, T helper cell, T cytotoxic cell, antibody secreting B cells and the dendritic cell. So these two major lineages forms a very important part of the immune system. Now, if we see and compare the embryonic and the adult hematopoiesis in humans, the hematopoiesis begins in the embryonic yolk sac during the first week of the development. So we can see the embryonic yolk sac where the hematopoiesis development has started taking place. Subsequently, it is taken over by the hemogenic endothelium and the fetal liver. But in the case of the adults or the grown-up adults, the differentiation of the hematopoietic stem cells majorly occurs in the bone marrow cells. So this is how the dependency and the growth and the development of the hematopoietic stem cells takes place. Now, in the immune system cells, one of the major classes of the immune system cells are called as the lymphocytes. So the lymphocytes are basically, it constitutes to about 20 to 40% of the body's white blood cells and 99% of the cells in the lymph. There are approximately about 10 to the power 11 lymphocytes in the human body. The lymphocytes are basically responsible for adaptive immunity, and they are given up certain attributes which includes the diversity, specificity, memory, the recognition between self and non-self. The lymphocytes have been broadly classified into three major populations. One is called as the B cells, the second one is called as the T cells, and the third one are called as the natural killer cells. So these all differentiate among themselves based on their function and the cell membrane components which is present on the top of it. Now, the lymphoblast cells proliferates and eventually differentiates into effector cells or into memory cells. So these memory cells are the cells which are responsible for the memory, and subsequently these effective cells are responsible to differentiate between the self and the non-self recognition. Now, if we take up the overview of the immune system cells, these are the three major populations. The B cells which are shown over here, so the electron micrograph we can see the small lymphocytes over there which can be a T or B cell, then the blast cell which is a T or B cells, and the plasma cells which are the B cells. So plasma cells are the differentiated and matured B cells which are secreting the antibody, while the T helper cells have got a specialized receptors present on the top of it, which are called as the T cell receptors. Along with the T cell receptors, there are certain other receptors which are also present, which are called as the CD4. While the cytotoxic cells or TC cells have got the T cell receptor along with the CD8 cell. So these are broadly the three major types of the immune system cells which are found in the body circulation. Now, if we see the fate of the antigen activated lymphocytes, so what happens is, as soon as the antigens interact with these kind of cells, the B cells, which are the naive cells, they get activated and subsequently they go into the cell division process. Now, in the cell division process, what happens is they differentiate or divide into two major categories. One is called as the effector cells, which are basically the plasma cells, and the other one are called as the memory cells, which remain dormant in the blood circulation for subsequent interaction. So whenever there is a second line of infection by that particular antigen takes place, these memory cells immediately recognize those particular antigens because they are specific for that particular antigen, and they neutralize that antigen from the blood circulation. So the effector cells are very important, as well as the memory cells are also equally important. Now, if we see the sites of development of these B lymphocytes, the B lymphocytes, the word has been designated from the site of maturation. In the birds, the B lymphocytes basically develop in the area which is called as the bursa of Fabricus in the birds, while in the humans it develops in the bone marrow cells. So bone marrow plays a very important role in the development and maturation of these B cells. Along with the bone marrow, if we can see the various lymphoid organs, which are the thymus, the lymphatic system is present, the liver, spleen. So these are all the various organs along with the lymphatic system, which plays a very important role. Now, the lymph cells, which are basically the B cells, these B cells are differentially distinguished by the synthesis and display of the membrane-bound immunoglobulin molecules, which serves as receptor on the antigen. Each of these are approximately 1.5 into 10 to the power 5 molecules of antibody on the membrane of a single B cells have been identical binding site for the antigen. So we can see an image of the B cell over here. On the top of the B cell, we can see the antigen binding receptors, which are nothing but the antibodies, which are very specific for that particular antigen. Now, what happens is, as soon as these B cells, they interact with the antigens, which can be the foreign proteins, viruses, bacteria, parasites or fungi, what happens is, as soon as they interact with them, they get activated and they synthesize or divide into the effector cells or the memory cells. Now, the effector cells had got a property of secreting the antibodies from the surface of it. So we can see the antibody secreting plasma cells over here. These plasma cells, because they have got activated, they secrete the antibody, and these antibodies are specific for the antigen. Now, this antibody binds with the antigens, and subsequently it facilitates the clearance of these antigens from the blood circulation or the lymphatic circulation. So this is how the interaction of these particular cells takes place, and this is how the other processes takes place. Now, if we see the human lymphoid system over here, the human lymphoid system, as we discussed, has been broadly classified into two major categories. One is called as the primary lymphoid organ, the other one is called as the secondary lymphoid organ. So the primary lymphoid organs are the organs where the initial development process of these leukocytes or the lymphocytes takes place. So these organs are basically the bone marrow and the thymus. So we can see the bone marrow, which is marked red in color in this pictorial diagram, and the thymus, which is marked red in color over here. So these bone marrow are the sites where the hematopoiesis start taking place. So hematopoiesis is basically the process by which the various red blood cells, platelets and the lymphocytes are synthesized. Now, these primary lymphocytes produces these ones. Subsequently, with the help of the lymphatic vessels, they move into the secondary lymphoid organ where they interact or trap the antigen. So now these secondary lymphoid organs are basically the sites or the areas where the interaction between the lymphocytes and the antigens takes place. So these antigens are subsequently neutralized and cleared from the blood circulation and the lymphatic circulation from those particular organs. Now, over here we can see the various secondary lymphoid organs, which includes the tonsils, the lymphatic ducts are there, then the Peyer's patches is there, spleen is there. So these all acts as the secondary lymphoid organ. Now, these primary and the secondary lymphoid organs are interacted or connected to one another with the help of the lymphatics or the lymphatic vessels. So we can see the lymphatic vessels which is circulating or present throughout the body of a human being. So these forms the basic part of the immune system in the humans. Now, the antibody term is basically a term which is given for a large number of proteins of high molecular weight that are normally produced by specialized B cells, which get stimulated or activated by an antigen and act specifically against the antigen in an immune response. So these are produced abnormally in the cancer cell. So these antibodies are specially produced by the specialized B cells which get activated when they get interacted with some antigen, which can be a foreign particle, virus, bacteria, parasite or any pathogen. Now, we will come into the structural details of these antibodies subsequently. Now, these antibodies, just we discussed, are generally represented by a structure of Y. So these antibodies on one of the end have got a antigen binding site. So these antigen binding site are very specific to which the antigen binds. So antibodies are proteins that are produced by the body to fight the intrusion of foreign molecules such as the toxins or other poison. The antibodies are designated to bind very tightly to their target molecules, which are basically the antigen. Now, if we see the representation over here, we can see there are different antigens which have got a diverse structural entities. Now, only the antigen which have got a complementary structure with the antigen binding site of an antibody or an immunoglobulin can bind to that particular site. So, an antibody will only interact with the cell that have the corresponding antigen. Antibody will not react with the cell that do not have the antigen. So this binding is more or less like the lock and key process, which is many times referred in the enzyme functioning. So the antibody specificity are very specific. The antibodies are very specific for a particular kind of antigen or foreign particle. Now, the antibodies mediate their effector functions with the help of the various pathways. The first pathway by which the antibody functions is called as the upsonization. Now, what happens is the phagocytes or the macrophages have got a property of endocytosis. Now, as soon as any antigen comes into the circulation, what happens is the antibody immediately recognizes these antigens because of the presence of the memory cells within the circulation. Now, these antibody, they bind to these antigens, and subsequently these antibodies bind to the antibody binding receptors which are present on the top of these phagocytes or the macrophages. Now, in the macrophages, these antibody binds, and subsequently the endocytosis or the engulfment process start taking place, in which the antigens is engulfed within these phagocytes or the phagosomes. Now, over there, the various acidic reactions takes place with the help of the compartmentalization, and these antigens are neutralized over there. The second property by which the antibody functions is called as the complement system. Now the antibodies, they activate the complement system, which is an array or series of proteins which subsequently binds to one another to form a membrane attack complex or a MAC complex. Now over here in the electron micrograph we can see the membrane attack complex which are formed on the cell membrane of the bacteria, fungi, and subsequently as these membrane attack complexes are formed, there are pores or cavities which are formed on the membrane which causes disruption of the ions, and subsequently the pathogen or the antigen dies off. The third property by which the antibody neutralizes the foreign particle, it is called as the antibody dependent cell mediated cytotoxicity, which is ADCC. It kills the cell. Now what happens is the antibody, as soon as they are recognized by the specialized cells, over here if we see the recognition is with the help of the NK cells, which are the natural killer cells. Now these natural killer cells recognizes the antigens which are bound on the target cells. Now these surface antigens are recognized by the natural killer cells and subsequently they neutralize these antigens. So this is how the reaction or the antibody neutralization takes place. Now one of the pioneer work in the antibody discovery was done by two of the eminent scientists. The name was Eldman and Porter. In the year 1972, they were awarded the Nobel Prize for the pioneer work they did for the discovery of the chemical structure of the antibodies. Now, in their award speech, what they said is there are probably more than 50,000 different antibodies which exist in the blood circulation. Each of them are reactive against a specific antigen. So we can see how specific the antibodies are. According to the antibody molecule, they appear in a shape of Y with a stem and two angles. Each branch is composed of one light chain and one heavy chain which are side by side, and the stem is made up of the remaining half of the heavy chain. So we will come into the structural details of the antibodies. Now the other works, pioneer works on the field of the immunobiology was done by three of these scientists, Journay, Kohler and Milstem. They, for their pioneer work, were given the Nobel Prize in the year 1984. They came up with the theories which concerning the specificity in the development and control of the immune system and the discovery of the principles from the production of monoclonal antibodies. Now, in their award speech, they said that the immune system is visualized somewhat like a gigantic computer where constant communications and regulations takes place in between the different components of the cells. The number of cells in such a network system in an adult human being exceeds 10 to the power 12, and the system has through its capacity to produce billions of different forms of antibodies and enormous inbuilt richness with regard to structural variation. So we can see how diverse our immune system cells are there. Now, if we see the basic structure of the antibodies over here, the antibodies, as we discussed, have got two major chains. One is called as the heavy chain, the other one is called as the light chain. So we can see the heavy chain which is marked green and blue in color, while the light chain is marked yellow and red in color. Now over here, each of the heavy chain and the light chain has got two major region. One is called as the variable region, the other one is called as the constant region. Now in the light chain, the variable region is designated as VL, while in the heavy chain the variable region is designated by VH. Rest of the region is called as constant region because the sequence of these constant regions in most of the antibodies remains same. The only difference in the sequence occurs in the sequence of the variable region, because this is the region which is specifically binding with the antigens over there. Now the variable, the light chain and the heavy chain are attached to one another with the help of the disulfide bond. Along with the disulfide bond, we can see two of the heavy chains are bound to one another with the help of disulfide bond, and this disulfide bond is present in the hinge region. Now, there are some of the fundamental experiments which were done in the chemical and the enzymatic method which revealed the basic structure of the antibodies. One of the first fundamental experiments and the breakthrough experiment which was done was with the help of the reduction and alkylation. In this one, the antibodies, especially the IgG antibodies, they were subjected to reduction process with the help of the chemical which is called as the mercaptoethanol. With the help of the mercaptoethanol, what happens is the disulfide bonds get reduced or the SH is formed over there. So the mercaptoethanol produce two heavy chain and two light chain, separating them from each other. So by this way, the scientists can designate that each IgG molecule contained two heavy chain which were having the molecular weight of 50,000 polypeptide chain, and two light chains which were having a molecular weight of 25,000. So the heavy chains were designated as H, while the light chains were designated as L. Now, the second experiment which was done was the brief digestion of the immunoglobulin with the enzyme papain, which produced 3 fragments. Now, with the help of these digestion, what happened is there were 2 identical fragments which were formed, which were named as FAB fragment or fragment antigen binding. Each of these fragments were weighing about 45,000 molecular weight. These were called as the FAB fragment because these contains the variable region which was specifically binding to the antigens, while the rest of the region which was found is called as the FC region or the fragment crystallizable region. So the other fragment was having a molecular weight of 50,000, which didn't had any antigen binding property at all, because it was found to crystallize during cold storage, so it was named in a such a way. So in the diagrammatic representation, we can see the immunoglobulin which has got the 2 chains which have got separated, which are called as the FAB regions, which contains the antigen binding sites, while one region which is called as the FC region, which is called as the fragment crystallizable. The third most important experiment which was done, it was done with the help of digestion with the pepsin. Now, pepsin is another different proteolytic enzyme which produced a single fragment composed of these 2 FAB-like fragments, and it was designated as FAB2 fragment, which binds to the antigen. So we can see over here with the help of the pepsin digestion, the 2 FAB regions were combined to one another, and from that part the cleavage or the digestion took place. While there was no FC region or the fragment crystallizable region which was recovered, because most of them were digested with the help of the pepsin. So in this way, with the help of these 3 major experiments, the basic structure of the antibodies was discovered, and it was found that it is made up of 2 major regions. One is called as the heavy chains, while the other one is called as the light chain. So these 2 heavy chain interact with the light chain, and subsequently these 2 heavy chains, they bind to one another with the help of the disulfide regions. So with this note, I come to the end of my first part of my lecture. In the second part, we will discuss about the various different types of the antibodies which are found in the human immune system. Thank you very much.
With this note, thank you, sir. Thank you so very much for giving us this productive session. Dear friends, you are requested to be with us, as we will be back after a short break and would be discussing more. Thank you for watching us. Hello friends, welcome back to the session. Friends, as you know that today we are talking on immune system, we are talking on how it works. In our previous session we discussed a lot on the topic and we believe that you might have gathered maximum knowledge through the previous session too. Now friends, in this session we are going to talk on different types of antibodies, and for the discussion on the topic we have with us in our studios Dr. Amit Bhattacharya. Dr. Amit Bhattacharya is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. So let's welcome our guest Dr. Amit Bhattacharya and let's try to grab maximum knowledge through him. Hello sir.
Thank you very much. So dear friends, in this part of the lecture I will be talking about the basic structural details of these antibodies, and subsequently I will talk about the various different types of the antibodies and how exactly they are important in our immune response and how they neutralizes the various functionings of the body. Now, the basic structure of the antibodies are many a times referred as the immunoglobulins. So, they are basically the heterodimers because they are formed by two different types of chains. One is called as the heavy chain, the other one is called as the light chain. So, there are two heavy chains and two light chains which are present. So, the light chains are having a molecular weight of about 25,000 molecular weight, while the heavy chains are about more or less about 50,000 molecular weight. Now, all these chains are made up of polypeptides or the amino acid. Now, each of the light chain is bound to a heavy chain with the help of the disulfide and various non-covalent interactions such as the salt linkages, hydrogen bonds, hydrophobic bonds, etc. Now, if we see the representation over here, this shows a basic structure of the antibody. So, on the top of the antibody, we can see the antigen binding site. These are the sites where the antigen binds, while the other parts are called as the constant region. The red part of the chain is shown as the light chain, while the blue part is the heavy chain. Each of these heavy chains are interacting with the one another with the help of the disulfide linkages. Now, the first 110 amino acid in each of the chain of the amino terminal region of the light chain or the heavy chain are varying greatly among the antibodies of different specificity. So, these chains are called, these regions are called as the variable regions. Now, these regions are called as the variable light or variable heavy in the light or the heavy chain. The regions of the relatively constant sequence beyond these variable regions have been dubbed as the C regions, which include the Cl, which is in the case of the light chains, while Ch, which is in the case of the heavy chain. The amino terminal portion of each of the light and the heavy chain is responsible for the binding of the antigen. So, the light chain sequences have revealed the immunoglobulins have got a constant and the variable region. So, we first discuss about the light chain sequences of the immunoglobulin. So, over here the amino terminal half of the chain consists of about 100 to 110 amino acids. This region was called the variable region. The carboxylic terminal half of this molecule are called as the C region or the constant region, and they have got two amino acid sequences which are called as the kappa and lambda. In humans, 60% of the light chains are kappa while 40% are lambda. A single antibody molecule contains only one light chain, never both. So we can see over here the variable region which is shown orange in color in the light chain, while the constant region is shown red in color over here. So the red in color region mainly can have either of the sequences. One can be kappa, the other one can be lambda region sequences. Now we come into the sequences of the heavy chain. Now the heavy chain, the amino acid terminal part, which consists about 100 to 110 amino acid, it shows great sequence variation, and this is the region where the antigen binding sites are present. So this region is called as the variable region, which is shown green in color over here. The remaining part of the sequences revealed has got 5 basic sequence pattern which correspond to 5 different heavy chain constant region, which are called as the mu, gamma, alpha, delta and epsilon. each of these 5 different heavy chain is called as an isotype. Now each of these classes can either have kappa or delta, lambda light chains which are present. So over here we discussed about the heavy chain sequences, which is the variable region which is shown in green in color, while the constant region which is shown blue in color over here. So there are 5 major sequences which are present, basic sequences which are present within this heavy chain constant region, which is called as mu, gamma, alpha, delta and epsilon. Now the heavy chain of a given antibody determine the class of the antibody. So if the chain is having a sequence of mu, then it is called as a IgM. If it is having a sequence of gamma, it is called as IgG. If it is having a sequence of lambda, it is called as IgA. If it is having a sequence of delta, it is called as IgD. And if it is having a sequence of epsilon, it is called as IgE. So based on the sequences of the constant region, the antibodies have been classified into 5 major classes. Now, in the careful analysis, it has been found that the first 10 amino acid sequences in the amino terminal part is responsible for the antigen binding, and it is present within the variable region of each of the light chain and the heavy chain. Now, each of these units have got certain domains which are present within them. So, each of these segments are having domains. Within the domains, the careful X-ray crystallography analysis have revealed that the immunoglobulin domains are further folded into a characteristic compact structure which are called as the immunoglobulin fold. So within these regions, there are presence of the domains which contains the immunoglobulin folds. Now, along with the immunoglobulin folds, there is another characteristic feature which is present to number of classes of antibodies, which is called as the Hinge region. So this Hinge region is basically present in the antibodies or the immunoglobulin IgA, IgD and IgG. So IgA, IgD and IgG has got a extended peptide sequence which is present within the constant region 1 and the constant heavy chain region 2 domains. So it doesn't have any homology with the other domains. So this region is called as the Hinge region. This Hinge region basically provides segmental flexibility to these antibodies. 2 prominent amino acids in the Hinge regions are proline and cysteine. Now we come into the various antibody classes one by one. Now the antibodies, the first antibodies or the prominent antibody class which is formed is called as the immunoglobulin G. Now the immunoglobulin G is the most abundant class of the antibody which is found in the serum. It constitutes to about 80% of the total serum immunoglobulin. Now the IgG molecule consists of 2 heavy chains and 2 kappa or lambda light chains. Now there are 4 IgG subclasses which are present. They are based on the differences in the chain sequences. So they are called as the IgG1, IgG2, IgG3 and IgG4. Now if we see the basic structural representation over here, the IgG over here is shown. So the blue color part is showing the heavy chain. So we can see a variable heavy chain which is shown over there. Along with that, there are constant regions which are there. Constant region 1, 2 and 3, and each of the heavy chains are interconnected to one another with the help of the disulfide bonds which is present within the Hinge region. In the red region is basically the light chain which contains a variable light region and the constant light region. Now the IgG plays a very important role in the immune systems of the humans. Now the IgG 1, 3 and 4, it readily crosses the placenta and play an important role in protecting the developing fetus. IgG3 is the most effective complement activator followed by IgG1. So we can see the developing fetus in the pregnant mother the IgG plays a very important role in protecting the developing fetus from the various infections. So IgG 1, 3 and 4 have got a property that it can cross the placenta and it can help the developing fetal to get protected from the various foreign particles. Now over here the various forms of the IgG are shown. The general structure of the 4 subclasses of the IgG are shown over here, which differs in the number and the arrangement of the interchain disulfide bonds. So IgG1 over here has got 2 disulfide bonds, IgG2 has got 4, IgG3 has got 11 disulfide bonds over here, IgG4 has got 4 disulfide bonds over here. So based on the disulfide bond, they have been further classified. Now the second important antibodies in the antibody classes is called as the immunoglobulin M. The immunoglobulin M, it accounts for 5 to 10% of the total serum immunoglobulin with an average serum concentration of 1.5 mg per ml. The monomeric immunoglobulin M weights to about 1,80,000 and it is expressed as membrane bound antibody on B cells. Now the IgM is secreted by plasma cells as a pentamer in which 5 monomers units are held together with the help of the disulfide bond. Now if we see the picture of the IgM are basically synthesized from the plasma secreting B cells over there. Now the IgM are generally present in the pentameric form. So 5 monomers of the IgMs are present. Each of them are interconnected to one another with the help of the disulfide bond, and these disulfide bonds are connected with the help of a J chain. So over here we can see 5 monomers of IgM which are interconnected over there. So that means that there are 10 antigen binding sites which are present in the pentameric form of the immunoglobulin M. Now the immunoglobulin M is basically the first immunoglobulin class which is produced in the primary response to an antigen, and it is also the first immunoglobulin to be synthesized by the neonate. IgM can bind to about 10 small haptens or antigen molecule. However, due to steric hindrance, only 5 or fewer molecules of antigens can bound simultaneously. So we have seen the pentameric structure. Due to the pentameric structure, there are presence of 10 antigen binding sites which are present over there, but only 5 antigens can be bound to these pentameric structure because of the steric hindrance. Now the pentameric IgM is more effective than the other isotypes in binding antigens with many repeating epitopes such as the viral particle and red blood cells. IgM is more efficient than IgG at activating the complement system. Now the next antibody we are discussing over here is called as the IgA. Now IgA constitutes to about only 10 to 15% of the total immunoglobulin in the serum, and it is predominantly immunoglobulin class which is present in the external secretions such as the breast milk, saliva, tears, mucus of the bronchial and genitourinary and digestive tract. So the IgG is present in the external secretions in the various forms. Now the IgA of the external secretion or the secretory IgA consists of dimer or tetramer. Each of these dimers or tetramers are interconnected to one another with the help of the J chain, which was also present in the IgM molecules also. Now the secretory component is about 70,000 molecular weight polypeptide produced by the epithelial cells of the mucous membrane. Now the IgA type over here is shown over here. So there are 2 dimers which are interconnected to one another with the help of the secretory component along with the J chain over there. Now the IgA molecule has been shown to provide an important line of defense against the various bacteria such as Salmonella, Vibrio cholerae and the viruses such as polio influenza. So IgA plays a very important role in protecting us from the various bacterial and viral infection. the breast milk contains secretory IgA and many other molecules that help to protect the newborn against the infection during the first month of the newborn life. So IgA plays a very important role in protecting us and the newborn from the various bacterial and viral infection. Now, the next in class in the antibody is called as the immunoglobulin E. Now, the immunoglobulin E, the potent biological activity of immunoglobulin E allowed it to be identified in the serum despite its extremely low average serum concentration. Now, the immunoglobulin E antibody are specifically mediated or present in the hypersensitivity reactions which are responsible for hay fever, asthma and the anaphylactic shocks. Now, this IgE molecule was first identified in the year 1921 by two of the scientists who injected a serum from an allergic person intradermally into non-allergic individual. So this is how exactly the serum was first identified. Now IgE molecule is shown over here. Now we can see over here in the IgE molecule there is no hinge region. So there is a variable region. Along with the variable region there is a constant region which is classified as the C epsilon 1, C epsilon 2, C epsilon 3, C epsilon 4. Each of these heavy chains are interconnected to one another with the help of the disulfide linkages. So IgE don't have any hinge region present to it. Now in the case of the allergy of asthma, IgE plays a very important role in the inflammatory reactions. Now we will see into how exactly these reactions takes place. So allergy is basically the food allergy which we will be concerned about, is especially against the peanuts or the peanuts, which includes the almonds, cashew nuts and the walnuts. Now the asthma is basically a chronic disease of the lung in which inflammation mediated by environmental antigens or the infections causes severe difficulty in breathing. Now what happens is, as soon as there is a foreign particle or a allergic food material or a foreign particle comes near to the B cells, these interact with the surface bound antibodies of the B cell. The B cells get activated and subsequently it results in the cell division to form the effector B cells. Now the effector B cells produces a large amount of IgE antibodies which are specific against this particular antigen, which is shown as a pollen over here. Now these antibodies come and bound to the molecules bound to the surface receptors of the mast cells. Now the mast cells have got the surface receptors which are very specific to the Fc region of the antibody or the fragment crystallizable region of the antibody. So these antibodies which are secreted from the B cell comes and binds to the mast cell surface receptors. Now as soon as they bound to these mast cell receptors, what happens is they bind to these IgE molecule, they bind to the allergen or the antigens over there. The allergen cross linkage of the receptor bound IgE to the mast cell, it induces the degranulation, causing the release of substances which are shown blue in color, which are basically histamine and the other substances, and these substances as they are released into the circulation, they mediate allergic manifestation. So over here we can see the allergen come and binds to the IgE molecule over there, which is bound to the Fc receptors specific for the IgE. As soon as there is a cross-linking of the allergen IgE and the mast cell takes place, the granulocytes or the granules gets degranulated, and these granules are released, which are basically the histamines and the other substances which mediates the allergic reaction, and the hypersensitive allergic reactions start taking place within the body. So these are the various types how which the IgE plays a very important role in causing the allergic reaction. Now the next type of the antibodies are called as the immunoglobulin D over here. the new class of the antibody, it is called as the which has got a serum concentration of 30 microgram per ml, and it constitutes to about 0.2% of the total immunoglobulin in serum. Now IgD, it together with the IgM, is the major membrane bound immunoglobulin which is expressed by mature B cells, and its roles in physiology of B cells is under investigation. No biological effector function have been identified for the IgD now. Now if we see the structure of the IgD is also a heterodimer which contains a light chain and a heavy chain. The light chain is shown in a red in color over here, which contains the variable region and the constant region, while the heavy chain over here is shown green in color, which consists of a variable region and the constant delta C, delta 1, C, delta 2 and delta 3 regions, along with the hinge region, which is bound to one, which helps the binding of the heavy chains to one another. Now, the monoclonal antibodies in the therapeutic studies or in the various diagnostic and the imaging studies have played a very important role. So, monoclonal antibodies have been produced or secreted for various specific antigens. So, the monoclonal antibodies provides a very useful tool as a diagnostic, imaging and therapeutic reagents in clinical medicine. Initially, monoclonal antibodies were primarily used as the in vitro diagnostic reagent. So, over here if we see, we can see the various toxins which are there, which is called as the ricin, Shigella toxin and diphtheria toxin. Now, one of the latest techniques which have been developed with the help of the monoclonal antibodies is called as the immunotoxin. Now, the immunotoxins, as the name suggests, it contains an antibody or immunoglobulin which is interlinked with a toxin which can be a ricin, Shigella toxin or diphtheria toxin. Now, the immunotoxin composed of a tumor-specific monoclonal antibody which is coupled with a lethal toxin and has potential value for therapeutic reagent. So, over here we can see a blue color antibody which is shown over here. Along with the blue color antibody to this, there is a binding of a red color toxin which have been attached to this particular antibody. Now, as these antibodies are very specific for the the tumorous cells, so what happens is, as soon as the person suffers from the tumorous growth, these immunotoxins, after proper checking and diagnosis, these immunotoxins are injected into the body. As these immunotoxins are very specific for these tumorous cells, they will locate the tumorous cells with the help of the specificity, and the variable region which is present on the amino terminal part of these antibodies, with the help of the variable region, they will bound to these tumorous cells. Now, the binding of the tumorous cells, as they bound to the tumorous cells, the toxin will subsequently inject its lethal activity into the tumorous cells. So, with the help of the immunotoxins, we can see how exactly these particular tumorous activity can be substantiated or decreased. Now, this is how the immunotoxins are functioning over there. Now, the initial clinical response to such immunotoxins in the patients with leukemia and lymphomas and some other have shown promising results, and the research to develop and demonstrate this safety and effectiveness is under development. So, over here an antibody which is bound to a toxin cell is shown over here. Now, as these immunotoxins are injected within the body, what happens is these immunotoxins, they go and bind to the tumor surfaces over there, and as they bound to the tumor surfaces, what happens is these antibodies binds to the antigens which are present on the surface of the tumors. Now, this binding takes place with the help of the antigen binding sites of the antibodies. Now, we discussed about the variable regions in the antibody. So, these are the regions which are specific for the antigen binding. Now, as these antibodies, they are bound with the help, bound to the toxins with the help of the various bonds, these tumor-specific immunotoxins are engulfed with the help of the various phagocytosis processes. Now, as they are phagocytosed, what happens is they go into the subsequent reaction process and they come into the endosomes. Now, in the endosomes, what happens is these toxins are released from the antibodies over there. Now, these toxins, as they release into the cytosols, they are very effective in blocking the various functionings over there. Now, suppose over here we can see these toxins are prominent in stopping the functioning or the translational processes or some of the important proteins which are necessary for the tumor activity. So, over here we can see the immunotoxins which have been injected into the body, and from the surface of it, it gets engulfed into the endosomes, and subsequently it moves into the various internal processes, and subsequently it affects the transcription and the translation processes of the tumors. So, the various types of the antibodies plays a various diverse roles over here, starting from the various activities IgM, IgD, IgE, IgA and IgM. So, over here we have discussed about the various antibodies such as the IgG, which plays a very important role in activating the complement system. So, how exactly the complement systems are activated. Now, subsequently, the activation, along with the activation of the complement system, how IgD protects the developing placenta or the fetal from the infection, because the IgG can cross the placental barrier and how it can protect. Then we discussed about the IgM, how exactly the pentameric form can protect us from the various infection. Then we discussed about the IgA, the secretory IgA, how it is present in the various external secretions such as the mother's milk and tears, saliva, and how exactly it is protecting us from the various bacteria, virus infection. Then we discussed about the IgE, how it is present in the hypersensitive reactions, and then we discussed about the IgD, and subsequently we placed upon one of the most important therapeutic use of these antibodies, monoclonal antibody, in the production of the immunotoxins, which are playing a very important role in the cancer research nowadays. So, with this note, I come to the end of my lecture. Thank you very much.
And we would like to thank Dr. Amit Bhattacharya for giving his precious inputs to the lecture. Dear friends, we believe that your feedbacks are important for us, as well as you can write your questions to us at info.cec@nic.in. We'll try to give answers to your questions when next time Dr. Amit Bhattacharya visits our studio. Till then, yes, keep watching us through our YouTube channel, and yes, keep writing us. We will be meeting soon. Till then, take care. Goodbye. Thank you, sir. Thank you once again.